Hello, everyone. It's an exciting chapter today on genetics called Patterns of Inheritance. The main learning objectives is to understand Gregor Mendel's work. So he's the pioneer of all the basic genetic laws. So we'll talk about his work and we'll talk about the two laws he proposed, law of segregation and law of independent assortment. And as we progress through the lecture, we'll be understanding several genetic terminology like genotypes and phenotypes and homozygous, heterozygous, and so forth. We'll also understand how we can use Punnett squares, named after the scientist, to predict the outcome of crosses like a monohybrid cross or dihybrid cross. So these are all Mendelian genetics. We'll also lightly touch upon non-Mendelian genetics in terms of incomplete dominance, codominance, polygenic inheritance, and so forth, and also inheritance that's inherited through the sex chromosomes. So it's called X-linked genes. Why is genetics important? We can ask that question. Genetics is so important because we are studying, it is the scientific study of inheritance or heredity and also of genes. So our knowledge of how genes move generation to generation and how genes influence a trait can be used in all these other fields in biology. In other words, genetics is the backbone of biomedical sciences, medical sciences, like gene therapy and so forth, molecular biology, where there's a lot of cloning and genetically modified organisms, pharmaceuticals like vaccine production and even insulin production, understanding of how a cell develops, developmental biology, huge applications in forensics, like crime scene DNA analysis, and how you can use DNA fingerprinting, and also inheritance to catch the perpetrator. And how old is genetics? There's data that shows, not data, cultures where there's fossil records that show that humans were domesticating and selecting and breeding crops like 5 to 10,000 years ago. So here's the wild type of corn. And through a series of selections, which is nothing but genetics to select for a trait, and then crossbreeding the favorable parents to get the corn of modern day times. So genetics has been around for thousands of years now. And here's some modern applications where, like in biotechnology, especially, they have transferred the glow in the dark gene, the luciferase gene, into other animals and to study inheritance patterns. So it's very exciting. It's a very exciting field of study. Most of the food that we eat today, almost everything, is genetically altered, either selected for or it's GMO crop. Gregor Mendel, who lived in the 18th century, is considered to be the father of genetics. Very interesting biography. You should read about him. He's an Austrian monk in Brno. He comes from a poor family, a farmer's family. However, I think his sister gives up her dowry money so that he can enter the abbey, where he becomes a father. And one of his passions was to study and grow pea plants. So this is his plot. You can still see it. There's a museum for Gregor Mendel. He's considered to be the father of genetics because he proposed the laws of genetics even before the microscope was invented, or mitosis was discovered. Meiosis was not discovered when he did it. So people didn't know that there were chromosomes. So that's why it's very impressive that he proposed all these laws of genetics. one of his main contributions is that at a time when people used to believe in the blending theory, that means, what is the blending theory? So father and mother, their genes will blend, and that's what you see in the offspring. So people believed blending happened, but it was Gregor Mendel that proved that blending doesn't happen. Inheritance happens as discrete units. Now we know that it's called as a gene, but at that 18th century, that time they did not know this. So his sample plant, his model plant were peas. So he was very lucky in that most of the traits that he studied were governed by one gene. So it was simple for him, and he got like very clear results. So he's applying the scientific method to see how traits are inherited generation after generation. He keeps meticulous records. He kind of applies statistics and mathematics to his data. So all of this is very unique and advanced for his time. The plant he chooses is pea plants. There are so many traits in a pea plant. There's 7 traits that he studies. The plant can either be tall or short. So there are many traits, 7 important traits he studies. And it has a very quick turnover time. That means the season, the planting to harvest time is very short. So less than 3 months. And it can either fertilize itself, it's called self-fertilization, or you can cross-pollinate it to make hybrids. So pea plants are very attractive in that way. So here you can see in the picture as to how, if you take the anatomy of a flower, you can see the carpel or the ovary, the female part, and the anthers will have the pollen or the male part. So in this brush, they will take the pollen and they will dust it on the ovary. And the male and the female will come together to form a zygote to set seeds. So you can either self-fertilize the flower, that means put its own pollen on the ovaries, or you can cross-fertilize it. You can take the pollen from a different plant and then you can fertilize the ovaries that way. So this plant, the flowers have both the male and the female sex organs in it. So that's a very unique feature. And here's all the traits that he studied. So the flower in peas can either be purple or white. The flowers can either be terminal, that means blooming on the top, or in the axis of a leaf called axial. The seed can either be yellow or green. The seed texture can either be smooth or wrinkled. The pod is either inflated or constricted, and so forth. So Mendel studies the inheritance of all these 7 traits. Let's see what happened. Before we go into Mendel's findings, there's a few terminology I want you to master. So when you say phenotype, it refers to the physical traits of the individual. That means what you can see. For example, if you look at yourself in the mirror, your hair color, your height, your skin color, your eye color, the shape of your nose, all of those things are phenotype. Whereas if you describe the genes that you have, it is called genotype, G-E-N-O, genotype. So for every chromosome in our body, right? Remember, like where we have 23 pairs of chromosome, 1 chromosome came from mom and 1 came from dad. If you have the exact same gene, it is called homologous. If it is different, like see capital B and small b, it is heterologous. Capital letter denotes a trait that is dominant. Small alphabet denotes a trait that is recessive. So these are genetic terms. Dominant, the trait that is expressed. Recessive is the trait that is masked. And to predict the outcome of a cross, this is called Punnett square. So you will see a lot of Punnett square in the latest slides. Again, the dominant genes are shown in uppercase alphabets and the recessive gene is in lowercase. And another word that is not shown here is the word allele. Instead of saying the other gene I got from dad or the other gene I got from mom, you would call that allele. So a Punnett square is a visual representation of a cross in order to predict the outcome of a cross. So here in this Punnett square, they have written the male gametes here and the female gametes here. And then you just cross multiply it, pull them down together. So if both are the same, if both alphabets are the same, it is called homozygous, capital B, capital B. So this is homozygous dominant. Small b, so this is homozygous recessive. Whereas heterozygous means it has one dominant and one recessive. So this is how gametes are depicted in a Punnett square. Gamete means the sex cell containing a specific gene. So the first thing that Mendel did was monohybrid cross. Monohybrid means he's only studying one trait. So it is a cross between parents that they differ with only one trait. So here, this is the parental generation. What you can do is you can self the flower for several generations and get a homozygous plant. So the parental generation is denoted as P. So he's crossing purple flowers with white flowers. And then he's growing the seeds from that cross. And the first generation, everything is purple. So the first thing is you're going to ask is, how come there's no white? Where did the white parent go? So if you self-fertilize the first generation and get the second generation, to everybody's amazement, he saw the white back in a 3:1 ratio, 3 parts purple and 1 part white. So this, historical monohybrid cross that Mendel did showed that it's discrete particles. It is not blending theory. It also showed the phenomena of dominance. So here purple and white is crossed and you get only purple. So in other words, the purple phenotype is dominant. So the same cross depicted in a Punnett square. So you have a homozygous dominant. So this has been solved for many generations in order to get a double homozygous. So you have a yellow parent and a green parent. And when they were crossed, the first generation is a heterozygote. When you self-fertilize the heterozygote, so you can put a Punnett square, put the gametes, capital Y, small y for male parent, capital Y, small y for female parent, and then just cross multiply each one. And you can see everywhere there's a capital Y, it will be a yellow seed pod. And only in the double recessive condition do you see a green pod. So from this, he deciphered the law of segregation, that for every trait, there are 2 alleles that will segregate or separate from each other when gametes are formed. So this is what we see in meiosis, like how in a certain stage in meiosis, the chromosomes will separate. It also indicates the law of dominance. In a cross of parents with contrasting traits, only one trait will appear in the next generation, and the trait that appears is called as the dominance. And here the phenotypic ratio is 3:1. 3 yellow to 1 green. So this is an example of a monohybrid cross when you're studying only one trait. If Mendel also studied 2 traits, and that is called as a 2-trait inheritance, or Mendel's dihybrid cross. So he's studying both the seed color, it can either be yellow or green, as well as the texture can either be smooth or wrinkled. So here's the parental generation. So the cross between the homozygous parents, here's the heterozygote. And then here's the Punnett square for a dihybrid cross. And the phenotypic ratio he saw here was 9:3:1. So 9 yellow and round, 3 green and round, 3 yellow and wrinkled, and 1 green and wrinkled. From this, he was able to decipher the law of independent assortments, meaning the alleles will separate independent of each other and they don't have any effect on each other. So that's why it's called law of independent assortments. They segregate or separate independently. So this we now know it has the basis for in meiosis. So you can see the metaphase plate here. And the chromosomes could either have been like this, or it can have been flipped the other way. And then one chromosomal set doesn't have any influence on the other chromosomal sets. So they are independent of each other. What is a test cross? So when you do not know the genotype of an organism, you don't know if it is a dominant trait for homozygous or heterozygous dominant. So then you will do a test cross. So a test cross is a cross between the unknown genotype and a double recessive genotype. And then you look for a 1:1 ratio to find if it is a homozygote dominant or a heterozygote. So Mendel was lucky that none of the traits he studied had an effect on each other. That means all the 7 traits he studied in peas, they were all found in 7 different chromosomes. They were not linked in any way or influenced each other. Genetics, however, is very complicated. It's not as simple as Mendelian genetics. So there are many variations to Mendelian genetics. One good example is incomplete dominance. So here, the offspring will have a phenotype that is in between the phenotype of the parents. So if you cross a red flower with a white flower, the first generation, it is pink in color. In other words, it's neither red nor white. So red is not dominant and white is not dominant. So that's why it's called incomplete dominance. And if you solve this in the F2 generation, you'll see a 1:2:1 ratio. 1 red, 2 pink, and 1 white. So that is the unique ratio that you will see in F2 generation. So this is a deviation from Mendel's laws. Incomplete dominance, also seen in snapdragons, which is a very popular garden flower. Another example of deviation would be codominance. And see, in Mendel, it was 1 gene, 1 trait. But now here's an example of many genes responsible for 1 trait. So that is called multiple alleles and codominance. Good example is the blood type that we see in human beings. So you can either be A, B, AB, or O. So we have 3 alleles for a single gene. And that's why, and none of them are dominant, they are codominant. So this is what leads to the 4 phenotypes of blood typing. So a person with blood type A will have genotype. So this is the immunoglobulin, RBC, and these are the carbohydrates that's stuck in it. So A, if your blood type is A, you have these A carbohydrates attached to the red blood cells. If you are a B blood type, you'll have B carbohydrates. So a person with A blood type can only take blood from another A type or O negative. AB has both, O has none. So that's why O is considered to be a universal donor, O negative. Remember that blood typing in humans is an example of multiple alleles and codominance. Multiple allele means more than 1 gene responsible for a trait. Here's another interesting phenomena that is not Mendelian. It's called pleiotropy. Here there is a single gene. So here's a chromosome. It's showing a single gene. The location of a gene in a chromosome is called as a gene locus. So when this gene Yeah, this is sickle cell anemia disease. So when that gene is mutated, it has a lot of bad effects on the body. So it's not one gene, one trait. Here's one gene having a lot of bad effects. It can affect the cardiovascular system in your brain. It can it can damage your lungs and so forth. It's a very bad disease to have. Because of one mutation and a change in one amino acid, the red blood cells will kind of collapse on each other to form the sickle shaped cell, which will stick together and it will clog arteries. It's a very serious inherited disease. Polygenic inheritance is talking about an inheritance where many genes are responsible for one trait. So they can be like the many genes can be spread apart in many chromosomes, and they are all responsible for one trait. For example, your skin color, right, as well as your height is polygenic inheritance. So 1, 2, 3, 4, 5, 6, 7, 8. So they here this Punnett square is talking about 8 possible alleles for the gene. So it's very cumulative. If you have all the tall genes, let's say you got 3 from mom and 3 from dad, to get a complete set of tall genes, then you end up being the tallest. So if you take the height of human beings, if you check in a population, you will see this bell curve. Most people will fall in the middle, that would be average height. But extremely tall and extremely short, you'll see in the tail end of the bell curve. So what is polygenic inheritance? Poly means more than one. So more than one gene is responsible for one trait. Example is your height as well as your skin color, your eye color too. The next non-Mendelian inheritance is the inheritance that can happen through the sex chromosomes, X and Y chromosomes. So sex-linked will describe a specific pattern of inheritance that can be traced back only through the X and Y chromosome. So here the red female, these are fruit flies, and it's a very good model system for studying genetics. There's a scientist called Morgan, and then he studied linked genes. If the gene is very close together, they kind of travel together. For example, red hair and freckles, right? So those two traits will go together because they are linked genes. So many of those studies were done in Drosophila. However, this picture is showing the inheritance that happens through the sex chromosomes. Because females have 2 copies of the gene, because they have X and X, even if one of them is mutated, they have a backup in the other X chromosome. Whereas X and Y, men do not have that privilege. That's why hemophilia and color blindness are more prevalent in men compared to women. Epistasis is a phenomena. Epi means on top of, stasis means to affect its expression. So epistasis means one gene in one location will affect the expression of other genes in another location. A very good example is, coat color in, cats and mice. Pigmentation of mice is shown here. So the wild type coat color, capital A, is the dominant, and it will have agouti is dominant when compared to solid color, little a. But if this gene, C dominant, is present, it will take over the color expression. So epistasis means one gene in one location having an effect on the expression of another gene in a different location. However, in the end, the phenotype not only depends on the genotype, but also the environment. So most of the human traits are a combination of both hereditary and your environment. For example, your height, right? How tall you actually end up being is because of your genes that you got from mom and dad, like 70 to 80% of it. But the next 30% comes from nutrition. your hormones, your sleep pattern, and your health and stuff like that. Here's the same hydrangea plant. This one is grown in a basic medium and then it turns pink. And here this is grown in acid medium and it's blue. Here we have acidic soil. So if you've seen hydrangea in Massachusetts, you'll probably see blue. Even though they have the same genotype, the phenotype is different. So it's a very clear indication of how the environment can affect the expression of a trait. And these are like interesting traits that you can study in the human body that you can look at later. And these are the most important points in this chapter. And I hope that was helpful.